Thanks, Hernan. Can you guys hear me? Is this working? Yep, okay. Well, it's wonderful to be back in Berkeley. It was an absolutely terrific day, very stimulating. And maybe I'll start with a little story about the origin of the BioHub. My job interview with Mark Zuckerberg. The original conception of the BioHub, which was their first science philanthropy, was that it was going to be some kind of joint institute between UCSF and Stanford. And I was involved in the planning, and eventually I got asked to be a candidate for the leadership. And so I was invited to Mark's house one day, and he just starts quizzing me, and I started quizzing him. And then ultimately I said, look, if you really want to do something with your philanthropy that lifts up the whole Bay Area, you've got to include Berkeley, because it's the third great university there. There's 3 great universities, and why would you leave out one of them? It was like, okay. And sure enough, that became such an integral part to the success of the whole program. It was awesome. But I think my Stanford colleagues have never forgiven me. Today, I'm going to talk about some work we did. In fact, the work that motivated me to get involved with that in the first place, and trying to do something bigger than a project in my own lab, and a large team science project. And it's aimed at answering this question of how do many cell types arise from one genome? And I'll tell you what I mean by that as we get a little further in. And I'll preface this by saying this is basically biology talk. There's a lot of physics under the hood, and I'll tip my hat to it as we go through. But just kind of relax your physics minds and enjoy the biology ride here. And we can dig in the question section into some of the physics if you want. So cells, we'll start with that. They're the fundamental quanta of biology. The smallest indivisible units of life. And this is a painting by a scientist and artist named David Goodsell, where he's taken kind of the protein structures of the various constituents of the cells, the proteins that do the work, the membranes, the genome which encodes sort of information about what the cell should do, and try to represent them in paintings. And this is an example showing kind of the density of a cell and all the protein machines. The stringy thing is the genome. And I realized this morning that this was a terrible choice of title for the slide because, in fact, cells do divide, and this painting is showing a cell dividing into 2 cells. But they divide on their own accord, and they're not divisible otherwise. The sort of past decade has been an amazing time in cell biology. It's been sort of revolutionary from the perspective of measurement technologies and what we've learned about how cells work, as both in isolation as part of an organism. This is going to be sort of one aspect that will be the topic of the talk today. So the genome, let's talk about that for a moment. As you might have heard, the genome is a molecule called DNA. It's got this famous double helix structure, and this encodes the information for synthesizing proteins. And the information is encoded chemically using a 4-letter alphabet. There you go, A, T, G, C. It's been sort of the last 20 years, the revolution of how to sequence this information and read it out, and I'll talk about that in a moment. But this is sort of the molecule of genetic inheritance. And as a sort of tip to the history of it, the structure of DNA was discovered by Watson, Crick, and Franklin. Watson passed away last month and had a very complicated story, if you've read the news around that, in his life. But their own stories, Watson and Crick, they wrote their own autobiographical books about it. And Franklin passed away tragically very young, but there's a beautiful biography of her, The Dark Lady of DNA. It's an amazing subtitle and captures a lot of the complicated dynamics around that work and how she was treated by her colleagues. But when the structure was first published, Watson and Crick would go around saying, we've discovered the secret of life. And I want to say, part of the lesson here is it's not all about the DNA. But that'll become clear in a minute. Let's just take a minute to talk about DNA by the numbers and sort of how you think about the genome. Every one of your cells has about 2 meters of DNA in it. So it's a very long molecule. And so if you work out that you've got some 37 trillion cells in your body you have enough DNA to go from the Earth to the sun and the back more than a couple hundred times. That's amazing, right? How does that work? It works because while DNA is macroscopic in one dimension, it's atomic in the other 2 dimensions, and it has a diameter of only 2 nanometers. And this is a movie we made in my lab years ago using optical tweezers to manipulate a long DNA molecule and tie it into a knot. This was sort of the first part of my career. I was really interested in single molecule biophysics. I spent a lot of time using DNA as a model polymer and studying things like forces and dynamics and topology and so forth. But it's sort of amazing that with optical microscopy, you can put a fluorescent dye in and you can see the molecule. Because it's macroscopic in one dimension, and there's enough sort of fluorescent signal to image that. As I mentioned, we have lived through a revolution of sequencing the information out of DNA chemically that was driven by, actually, DNA sequencing as a process going from an analytical chemistry technique based on separations to a biophysical technique based on fluorescence microscopy and imaging. And that vastly increased throughput, lowered cost, and this is sort of a dated slide showing sort of cost going down over time and amount of information going up. And I was involved in that. We developed the single molecule biophysics project in the lab became a single molecule sequencing project. We built a sequencer, sequenced my genome, and that's what kind of pulled me into this world of genomics. So how does the information for the genome get used by the cell? The so-called central dogma, which was an ill-chosen phrase, was the model, mental model for how information transfer was used. Information processing was done for protein synthesis, where it's copied from DNA to RNA, and then through transcription, and then translated by the ribosome into protein. And so this RNA intermediate ends up being a very important part of the story today, because as you'll see, the genome is the same in every one of your cells. The RNA is different. Proteins are different. Proteins are hard to measure. RNA is easy to measure. So RNA becomes a really fantastic proxy for measuring the state of any cell. Finally, I'll just say that, all the physics criticism that biology is just a bunch of stamp collecting and, there's no underlying theories or unity to it. There are many aspects of biology which do have an underlying unity. They're common and universal across all organisms. And that's what Monod had in mind here with this quote, anything true of E. coli must also be true of elephants, that by studying simple model organisms, E. coli being the sort of hydrogen atom of biology, you'll learn a lot about other organisms. And if you want to sort of read some of the history of that, this is a tremendous book called The Eighth Day of Creation by Horace Fleehan Judson that goes through discovering the structure of DNA, discovering central dogma, learning how genes are regulated, solving the structure of proteins, and it's an amazing, beautiful story. Just really terrific. Okay, so why is the genome not the be-all, end-all? People, when they speak loosely, say the genome is the blueprint of the organism. That's actually not the way to think about it. And the example is what I mentioned earlier. Every cell in your body has the same genome, but a heart cell is different than a lung cell, is different than a brain cell. They're very, very different in what they do. But they all have the same genome, so it's not the blueprint. I think a better sort of metaphor is to think about the genome as a parts list. It's got a bunch of parts. Every cell type chooses some of those parts, and that's how they choose their identity. It's not every gene expressed all the time. And so there's this fundamental question of understanding how do cells decide to use which parts and in which situations. And hence the title of the talk, how do many cell types arise from one genome? And should you think about the genome as like a computer code, which is deterministic and controls cells and tells it what to do, like a piece of software? Or does physics matter? Fluctuations matter. Every cell is a single molecule experiment at some level because there's 2 copies of the genome in human cells. And so there's many open problems. The central dogma was just about understanding information flow and protein synthesis, but there's many, many other questions now. And like the one that I've been obsessed with is, how do you predict the cell types of an organism from its genome? There's no first principles way to do that right now. If we were to find a new species and I gave you only its genome, you can't predict what the cell types are. It seems like you should be able to do that, right? I think it's a reasonable hypothesis that the genome contains that information, but open problem. And so we began to work on this experimentally. And sort of the thought experiment was, what if you could enumerate every molecule in a cell? Forget about where they are, the locations, the structure. Just make an inventory, a list of every molecule in the cell and how many copies of it were there. Would that be a useful sort of estimate of cell state and cell identity? I think most people probably think yes. It's obviously a very difficult experiment to do, but there's a version of it that actually turns out to be tractable, which is to count every mRNA molecule in a cell. And is that a useful proxy? And I hope I'll argue and convince you, I will argue, and I hope I'll convince you through the course of the talk that that's a true statement. And single-cell transcriptomics has become the super hot, buzzy field in biology recently, and I like to always remind people of that old vaudeville joke that it takes 20 years to become an overnight success. There's a lot of things in science that are like that, and single-cell transcriptomics was 30 years in the making. It really started right after the invention of PCR, which was the first biochemical amplifier, and people began to think about how to amplify all the RNA molecules in a cell. And in 1990, that was done. That was just a couple of years after PCR was invented. And it's a version of an amplifier that lets you amplify the mRNA. And actually, two folks did it independently in that year and published it, but they couldn't read out the complexity. They could amplify the RNA and have this test tube now with lots of complexity and lots of copies of what was in the single cell. But they couldn't read it out. And so they were probing it just sort of one gene at a time. And it was just sort of the first step. And it took another decade and a bit to work out ways to read out that complexity. And that was done through the use of microarrays, which were sort of molecules patterned on a surface, and you could measure hybridization by sequence binding. And there are 4 papers that year that did it in a different ways, from thousands to tens of thousands of genes in an experiment. And then eventually, sequencing got powerful enough to read it out by directly sequencing the material. And that went through the revolution that I mentioned earlier. And so those were sort of most of the key steps along the way. And the only thing that was missing by this point was how to do it at scale. These were all sort of bespoke experiments with a handful of cells and just very small numbers. And that's sort of where I got involved, because the other part of my career had been developing microfluidic automation, so miniaturized fluidic circuits to automate biology. And we began using those to sort of automate this process of single-cell transcriptomics and do it in a very highly parallel way. And so we had been using these tools for many other things, and I had actually a super awesome collaboration with James Berger here for many years before he moved on from Berkeley to Hopkins. But we had this awesome sort of moment where my student came up to James's lab and did more experiments in a week than his best postdoc had done in a year. And then we sort of knew that this microfluidics was going to be useful for something. And eventually it became useful for single-cell transcriptomics, and a company I founded called Fluidigm built the first commercial device to enable people to do this. It was called the C1. It was launched in 2012. And there was a lot of interesting physics behind how you make these devices, how you use them. There's sort of basic fluid physics I'll talk about on the next slide. There are many interesting engineering aspects to how you think about these like integrated circuits, sort of a lot of solid mechanics around it, figuring out how to manufacture at a scale. At some point, the company realized they had made more plumbing than all of California, Texas, and New York had together at the macro scale. And that was a whole adventure on its own. This was used by many hundreds of labs around the world, and it kind of broke open the field so that any biologist could start using this technique. And it just only increased people's appetite for more. I mean, the chips here did the amazing throughput of 96 cells in one experiment, and that was transformative, but people wanted more and more. So the next generation of technologies came, also based on microfluidics we developed in the lab, and that's a way to make microfluidic emulsions, sort of oil and water mixtures, and we discovered quite some time ago there's a very interesting instability. If you have 2 channels come together and you have oil in one and water in the other, they're not miscible, and sort of just there's an interesting dynamic instability that would butt off little droplets. We explored the physics of that. That eventually got turned by others into tools for single-cell transcriptomics that let you do thousands of cells in an experiment. That became widely used. Now we're on to a 3rd generation of technology in the field, which is another order of magnitude more. And it's just been sort of amazing innovation. So with that as background, kind of the way you would do an experiment to do single-cell transcriptomics is you take your tissue, you cut it out of the animal or the donor, you enzymatically digest it, so there's single cells in solution. You put it in one of these microfluidic devices where they get encapsulated, knocked open, amplified, barcoded, so every cell has a different barcode. Then that all gets read out on a sequencer, and then you have this very complex high-dimensional data set to sort of compute on. So every cell is represented by a 20,000 dimensional vector. Okay? The human genome has 20,000 genes, and each sort of entry in that vector is a different gene, and the number is the number of mRNA molecules of that gene that were expressed in that cell. Okay, that makes sense? So that's the representation of every cell, and you can do this for thousands to millions of cells. And it's a very interesting problem how you compute on that, and you've maybe seen pictures like this before. Every dot here is a different single cell in that 20,000 dimensional space. They've been clustered in the 20,000 dimensions, and then there's many interesting algorithms and computational approaches to do sort of dimensional reduction, kind of like PCA, but nonlinear versions of it that let you understand the relationships in sort of a two-dimensional plane. Okay, that's the approach. I started the BioHub and got involved in the Zuckerberg philanthropy because I wanted to take these techniques, we'd spent decades developing them, and apply them at a large scale to an entire organism and build a cell atlas to do that. And so we did that. That was the first 5 or so years of the BioHub. And we started with mouse and then got involved in doing a fly atlas with the international community. Then we went and did human, and we also did lemur along the way as well. These were sort of terrific team science projects. Each paper has like 150 authors on it. It involved people at the BioHub and at all the 3 universities. And it was a lot of fun to figure out how to do that. Especially initially with the mouse, which was the first one, we kind of took the Tom Sawyer approach. We didn't fund anybody to do it, but we recruited tissue experts different labs that were experts in the biology of each tissue. There were 18 for mouse. And we said, do you want to be part of this? And they all did and signed up. We paid for the reagents and the sequencing, but everyone volunteered their time. And it was kind of like, they were so excited about getting a first look at this kind of data for their system. And it just was a great sort of fun thing. So with mouse, we started, it actually came out of a conversation with Tony Whisker-Ray, who had stopped by my office and was interested in using RNA analysis to look at aging in mice. And I said, well, why don't we just go do single cell measurements of that? And so we did. The challenge was it's hard to get an old mouse because they die. And we wanted to work to see really what happened with old mice that were like the equivalent of human centenarians. And so we knew we wouldn't have a lot of those mice. And so we worked very hard to develop ways to get multiple tissues from the same individual mouse, 18 different tissues, and process them all in parallel. And that required an enormous amount of sort of coordination and teamwork. And that became sort of one of the sort of watchwords and things we became known for in doing this work. That became very helpful as we moved on to other organisms like human. And 2 of the key people in this were young group leaders of the BioHub, Spiros Dermanis and Angela Pisco. So what does it look like? This is the 3-month time point here. As I mentioned, every dot is a different cell. After we got the data for each tissue, the tissue experts went and analyzed it all by hand. And they hand annotated every cell about what they thought the cell type was. And this became the largest sort of human annotated data set for the field and a very widely used reference. And we could do things. This was a very simple demonstration early on where we clustered all the data together in the high dimensional space. And then here it's colored by tissue. And we didn't tell the computer anything. It only had the numbers and the vectors, and that was it. It wasn't taught any biology. And you can see that most of the clusters are solid colors, which are different tissues shown down here. And we've got the names written on from the human annotation expert later, so you can see that they're reasonable cell types. Some of them are multicolored, and those are cell types that are shared across tissues, like B cells, which are immune cells, or endothelial cells, which are also found in many different tissues. And so here, that's showing that the computer was able to discover the shared aspects of these cell types, even though they came from different tissues in the animal. And by analyzing the gene expression profiles, we began to understand things like how you could potentially reprogram these cells. I won't dwell on that for these purposes. Each tissue is composed of many different cell types. And traditionally, before these techniques were invented, you would just grind the whole tissue up and measure RNA of all the cells mixed together. And that had value for certain things, but obviously not for all things. And kind of the analogy it's often used is making a smoothie. You can mix a lot of fruit together and study the kind of mixed effects, or you can study each one separately and learn about each fruit individually. And that is essentially what happens with this approach. And there's a very kind of clear example of that, I think. If you're looking at a cell type or tissue or any kind of sample under two different conditions, healthy, diseased, old, young, and you sequence into it and you see certain genes are changing their gene expression level. If it's a bulk experiment, there could be two explanations for that. One could be that their gene expression level is changing in each cell that it's expressed in, or it could be that more cells are expressing that gene. And from a bulk measurement, there's no way to resolve those two things. But you can resolve that with a single cell measurement, and we went and systematically asked about that. Here for, this was an aging question, difference of gene expression. What was driving changes in gene expression with age? Is it the number of cells or the effect size? And honestly, most of the time it's both. Both were contributing. And those are the first time we could deconvolve questions like that. We then went on to try to manipulate aging and look to reverse aging. There's a very clever approach called parabiosis, where you suture together two animals so they share a blood supply. And that had been used for a number of years to study the effects of aging. You connect the blood supply of a young mouse to an old mouse. The young mouse ages more quickly. The old mouse reverses aging by certain gross physiological measurements. And we decided to look at that at the single cell level. And I thought we were going to kind of refute the whole field and expose it all as a fraud. But I was wrong. I mean, it turned out there were actually real measurable effects, I believed, in the end. And it was sort of a fascinating study. And if you're interested, here's the reference. So let's talk about human, which became the really big project, the thing we were hoping to do from the beginning. We call that the Tabula Sapiens. And that was really led by a fellow named Bob Jones in my lab, a senior research scientist, and several other folks. You may recognize this guy, former colleague from Berkeley, and Julie Saltzman, Mark Krasnow, and so forth. I think I'll just jump. It was about 500,000 cells, the first version. This is sort of what it looked like. Each row is a different tissue. We aimed to get 24 tissues. Each column is a different human donor. And the entry here is the number of cells from that tissue from that donor. And you can see it all adds up to, in this case, 1 million. So this is the first chapter. This is the second chapter, which is not published yet. And we see roughly 700 cell types from the expert annotation. How do we do this? It was through an amazing collaboration with an outfit called Donor Network West. They are a nonprofit organ procurement organization who help connect organ donors to people in need of transplant. Oftentimes, people who have consented to be organ donors will also consent to have their tissues used for research. So things that aren't transplanted can sometimes be used for research purposes. We began to work with them to do this. And this was by far the most complex thing they'd ever done. And it was just amazing. Typically, the way it would work is we get an email from them or a call saying, we have a donor who's on life support, brain dead. We're going to take them off the life support in a couple of days and try and transplant some of the tissues. Do you think this is suitable for your project? And we'd look at the medical records and make a decision of whether or not we thought it was a suitable donor. And if so, then the surgeons would work after the transplant tissue is taken out on the rest of it for us. Somehow, it always seemed to happen at night. And these were really heroic experiments that, usually 24 to 36 hours. And surgeons would take out the tissues one by one. They'd be sent in ice buckets to the various labs, or they were processed. Typically, for those of you who think about this stuff, 50 10x libraries and 82 SmartSeq plates, and just absolutely amazing work from the students and postdocs and staff members who would do this. And my job here was to deliver pizza. I wasn't actually at the bench, but I would try to help out. And this gives you a sense of sort of the scale of the project and the folks who were involved. So what did we learn from this? Let me give you a couple of examples. And I'll come back to the secret of life. One of the stories in the 8th Day of Creation is about the so-called second secret of life, which Jacob and Monod discovered that some genes control other genes. I remember I raised this question, not every gene is expressed in every cell. Some genes control other genes, that's called regulation. And the way that's done is there are certain proteins which will bind to the genome at certain sequences, and they control whether genes are turned on or off. And broadly speaking, they're called transcription factors, and they control whether RNA is copied from the genome and then turned into protein. And it turns out there's, in the human genome, estimated to be about 1,622 transcription factors based on actual study of them and predictions based on their sequences. And we looked into our data set to see how many of those were expressed, and we found all of them. That was pretty cool. So then we began to study, well, which transcription factors are expressed in which cell types. Some are very ubiquitous, some are very cell type specific. We used a statistic called tau to measure that sort of specificity. Tau of 1 means only one cell type, and tau of small numbers close to 0 means lots of cell types. And so you see there's kind of 2 peaks in the distribution. And if you focus on the ones that are on the right peak that are very cell type specific, it looks sort of like this. Here, each row is a different cell type. Each column is a different transcription factor. And you can see that by and large, for these very specific ones, they're only expressed in 1 or a handful of cell types. And as we look at cell types that are very well studied, we see transcription factors that make sense with respect to the literature. But now we also have this data for cell types that are not well studied. And we sort of have it all mapped out in great detail. And this, as we were doing with mouse, provides an amazing recipe to think about reprogramming cells from one type to another, which has many potential applications in developing cell therapeutics. Here on the right, you see the opposite. These are the ones with low tau, and you can see they're expressed ubiquitously across all the cell types. Let's talk about those for a minute. This is now the data sorted by tau, and so these are the ones that you saw on the left. And what we see in the ubiquitous ones are many very well-studied transcription factors that are known to be involved in processes that every cell does, no matter what the cell type. Things like cell division, stress response, metabolism, and you'd expect to see that, and those are things that are known. That's good. But we also found some transcription factors that had never been characterized before. They're just hypothetical ones based on their sequence. And you can see them, many of them zinc fingers. They've got names that start with Z and F and then a number. And just amazing after, what is it, a century and a half, century is called a century of cell biology. There are ubiquitous transcription factors that every cell uses that have never been characterized. And so this provides, sort of a direct sort of route to studying some, I think, very important ubiquitous biology, and we're very excited about following that up. Other story I want to talk about has to do with senescence and aging. So there's a fellow named Leonard Hayflick who discovered something called the Hayflick limit. And there was a debate in the first part of the 20th century about whether cells are immortal. Could your cells divide forever or not? And there were a bunch of literature, people claiming that they could make cells last forever. Turned out that was wrong. The experiments were contaminated. Hayflick discovered he did it right, discovered cells are not immortal. They can only divide about 50 times or so before they just stop. And they enter a different state where they don't divide anymore. He called that state senescence. And we now associate it with aging and such forth. And you could calculate from this, if you're only allowed 50 divisions per cell, there should be a maximum theoretical human lifespan. You can do the numbers on that, which he did, came up with 120 years. And that seems to have held up. Very few people surpassed that, if any. Hayflick himself died last year, and someone at the New York Times had a lot of fun with the headline of his obituary, which is Leonard Hayflick, who discovered why no one lives forever, dies himself at age 96. And there's been an enormous amount of effort over the past decades in understanding the molecular details of senescence, what drives it, what do senescent cells do. They contribute to many aspects of aging and disease. And we realized that we had in the Tabula Sapiens the largest ever collection of human senescent cells from actual living human beings, not cells that have been artificially manipulated in a dish or a lab plate. We went through and found, I don't know, it was like 50,000 senescent cells using sort of the classic markers, appearing at roughly the frequency you'd expect. And we had not only these cells, we had the full 20,000 dimensional gene expression vector for each of them. So we could start asking questions about their transcriptomes and their biology. And the first question we decided to ask is, are all senescent cells senescent in the same way? I mean, kind of like the Tolstoy, right? And it turns out not. And so if you ask, is there a common gene expression signature that defines all of senescence, that all cells use? There was not. Only the ones we use to define senescence. There's a circular argument about P16, CDKN. Let's put those aside. But if we looked for differential gene expression and asked which genes were differentially expressed between different types of senescent cells, it was like, a fraction of cell types for any given gene. And we're seeing many classical markers pop up here of senescence that have been studied, some that are not so well studied. And that was kind of the sort of a very interesting discovery. And then we began to ask, okay, is every senescent cell different than every other senescent cell? That's fortunately not the case. And in fact, there seem to be several broad programs, about 16 different gene expression programs that are used in different ways across half a dozen classes of senescent cells, many of which are shared across tissues. And so here you can see, again, these are now here shown by cell type, here colored by cell type, here colored by tissue. And so you can see that these different types of senescence are found in different tissues in a way that's sort of interesting. Some of them are pure cell types, immune cells, endothelial, epithelial, B cells, the ones that do tend to be shared. So there's sort of a ton more to do that we got out of this. These are resources that are essentially companions to the genome. And we think about it as a phenotypic companion to the genome that teaches you how the genome is used in different cell types. There's other biological questions about whether the transcriptome is a reasonable proxy for physiology, which is how cell types were classically defined. That's a whole other talk, but it turns out it's remarkably good. We spent a lot of time thinking about that. But what I want to do now is tell you a bit about how we're thinking about integrating all that data together into a single atlas and what that means. And at CZI, we built a database to do that, to collect all the single cell data from the literature and many from groups we funded, not just the work at the BioHub, but we had grant programs all over the world for specific tissues. This all got pulled into a database called Cell by Gene, which anybody can explore. It's got an easy graphic user interface. You can also download all the data. It's now up to more than 100 million cells. And it's a really neat resource. And yet, it's just a giant table, right? It's a list of cells and genes, a giant matrix. And you don't necessarily get a lot of insight out of that. So we began to think about using AI approaches to analyze and synthesize the data and all that, and also to find ways to integrate data across species. Everything in Cell by Gene is human and mouse, but there's other data from other organisms. It's interesting to think about ways to compare them. So I think this slide is now probably obsolete. We all know we're in this amazing revolutionary era of AI. These are fun I had asking DALL-E, which is now obsolete, to make things like a painting of all the proteins in a cell, or I want to make a cell atlas. And, amazing things. And how are those models trained? Let's focus on the language-only ones, perhaps. If you use ChatGPT, you're trying to predict text. You cover up some portion of a text, try to predict the rest of it. And you could do the same thing with the gene expression data, right? You could take a given cell's 20,000-dimensional transcriptome vector, cover up some of the genes, and try to predict them based on what the expression levels of the other ones. And so with Yuri Leskovich, we decided to try to do that and built a model, which we called the universal cell embedding. Referee number 3 hates that, so we're probably going to change the name. Might be the general cell embedding or something like that. But it's, physicists always get freaked out by the number of parameters and models like this. I've gotten comfortable with it. This is like the giant adjustment to make. 650 million parameters. You can calculate what, 20,000 dimensions times 100 million cells is, and you're probably underfitting, so it's okay. This is, the size of the compute we did. Trained it about a month. So we did it in the way I just described. You cover up 20% of the data and try to predict it from the rest of the data using transformers and such forth. We also did a lot of work to try to make it work for any organism, any genome, any organism. It's not dependent on the underlying genome, using something called protein language models. And that's a way to create, the technical folks, an embedding space where any gene can be embedded, and it doesn't matter what the genome was, and you're not referring back to the genome. And so it allows you to compare genes from any species in a way that's sensible and meaningful in distance without having to refer to the genome. And so we used those embeddings to represent all the genes and then did the gene expression data. And so that was Yuri's idea, and it was a really super powerful idea. And so we can use the embedding space that comes out of this now to measure distances between cell types. And we trained the model on sort of 7 or 8 different species: human, mouse, lemur, zebrafish, pig, macaque, and frog. And you can see it sort of here, colored by cell type, colored by data set, colored by tissue, colored by species. And so vast majority of it was trained on human. Next most common is mouse, and the other ones were a small fraction of it. So bear that in mind as a caveat. And so having built the model, we got interested in how good is it and how well does it work on data it hasn't seen. This is sort of known as zero-shot measurements. So we used a held-out data set. We only trained it on the human part on the first chapter of Tabula Sapiens, not the second. That was unpublished. We kept it out aside, no chance for data leakage. So then we embedded that second chapter into the model and asked, where do the cells go? And they go exactly where they're supposed to relative to their counterparts from data of the first chapter. And so with the data set it's never seen before of human, the model did a very good job of embedding those cell types and putting them in the right spot. We then began to ask, well, what happens if you give it an organism, a species it's never seen before? What will it do with cell atlas data from that? And so here, again, data from, Green Monkey, the lymph node experiment. This is published by another group. We didn't include it in the training data. We embedded it in the model. We asked what cell types are these cells closest to? And so these are the predicted cell types based on the human annotations. This is how the authors of the paper had annotated them. And the agreement is reasonably good. So the model can perform on species it's never seen before, which is like of enormous potential value for evolutionary biology. And I think this is going to be a very important approach for that field and time. And you can ask, has the model learned any biology? Anything about the relationship, the biological relationships between cell types, also known as lineage relationships. And remember, the model wasn't trained on any biology. It was only given a matrix of numbers, just gene counts, nothing else. No gene names, no literature about biology, no cell type annotation. So this is really a question, like we referred to earlier, about what are the relationships in the originally 20,000 dimensional space. The embedding space is smaller between cell types. If you ask about distances and you start to build a dendrogram, you see that it kind of makes sense. So T cells, which are different, they're immune cells found in different tissues and different flavors of them, they're all very close together. And they're close to other immune cells, but they're farther away from muscle cells and endothelial cells. And it's sort of, the distances within the space are meaningful with respect to the biology that we understand about the cell types. And you can ask that in an even more general way by saying, let's compare, there's something called the cell ontology tree. And so somebody's gone through and made a distance, sort of tree of all the cell types and how they've been labeled in biology and what their distances and how we believe they derive from each other, their relationships in the development of the organism. And so you can compare those tree relationships from the literature and knowledge of biology between parent, child, siblings, cousins, distant relations, and so forth, and ask, for any given relationship, what's the histogram of distances of cell types that satisfy that relationship? And the more distant the relationship, the more distant the distribution of distances in the embedding space. And so they're getting further away in embedding space as they're further away in biological relationship. So that also is very powerful, to my mind, emergent behavior from the model. So we've got great hopes that this will be used for many different applications going forward. We've already gone on and built a second generation model at CZI called Transcriptformer, which is trained across a dozen different species. We've been a lot more disciplined about the training. There's a preprint for that up. And it's going to be a very interesting area going forward. I got a few more minutes, right? Okay. So in the end, I just want to talk a little bit about some of the applications to human health where we see all this going. We went after this as a very sort of basic science type question, but in biology, basic science and sort of human health disease applications are often two sides of the same coin. One of the things I've been very interested in my career has been Hernan mentions in the introduction, developing blood tests to replace invasive biopsies. And I've done this in areas from pregnancy to organ transplant, infectious disease, cancer. What happens is the various tissues of your body will contribute RNA into the blood, and it all gets mixed together. So if you take the blood, purify, and sequence that RNA, you're getting signals from a mixture of many different tissues and cell types. And there's now a question, sort of a logical one, can you use this Tabula Sapiens to deconvolve all those contributions, treat it like an inverse problem? And it turns out you can, and a very talented student named Sivan Raporian did that in the group, and she worked out what the various cell type contributions are, many from the blood, those are the dominant ones because that's the proximal bit, but also from many of the solid tissues, and then worked out a nice way to invert these things, and then look at how cell type representations were changing in disease. In the case of liver disease, you're seeing signals go up so these are metabolic diseases of the liver. These are signatures that are cell type specific from the liver, meaning more cells are dying. Here in chronic kidney disease, this certain cell type within the kidney is going the other way, but measurable signal differences in health and disease that are very cell type specific. So we're very enthusiastic and feel that this is a good start for understanding that. We're also using the Cell Atlas to look at developing the new therapeutics, or using the techniques we use to make the Cell Atlas to develop new therapeutics. This actually is an example that goes back to the earliest days when we were first working out these techniques. Mike Clark and I were collaborating to use them to look at tumor heterogeneity in cancer and try to identify the molecular details of different subpopulations of a given tumor to find new drug targets, specifically to try to attack tumor-generating cells like cancer stem cells. And we founded a company, we published some work on that, founded a company in 2010. It was acquired in 2015. That company got 3 drugs, small molecule drugs, into humans. And we're still trying to figure out how well they work, but there's some very promising data along the way. And this was kind of a first proof of principle about how to use these single cell techniques to find new drug targets and to make drugs against them. People are using the Tabula Sapiens in a similar way. When they found a drug target, they're looking to see what other cell types it's expressed in. That's called on-target toxicity. So there's a way to understand potential toxicity using the Atlas. And we've had many people use it for that. That's been very cool. Final example is one around food allergies. Derek Crute, who's a student in my group, interested in working on food allergy projects. Kari Nadow, who's an allergist at Stanford, an immunologist. We began using the single-cell Atlas techniques to look at the blood of children with food allergies, in this case peanut allergies, and we're able to find the cells that are making the bad antibody that causes the allergy, sequence that antibody, and then clone and express it so we could make the protein and study it. And found that these antibodies that cause the peanut allergy were shared across different people. They had an interesting clonal relationship in the sense that evolution found the same solution twice, like 2 different people making the same antibody. That never happens, and down to the sequence level. And those ended up being very interesting antibodies, super mutated, and they converged on similar sequences. And we realized we could re-engineer them. We could tune their affinity to the offending allergen, and we could re-engineer them so that they would be not harmful but beneficial. Founded a company called iGenix to do this at scale. Here's like a similar experiment with thousands now of antibodies and sort of showing this convergent evolution happening again very broadly. And as the antibodies were re-engineered to be able to bind the peanut but not cause an allergic response, we administer them to mice, and they provided protection to the mice against allergic response. And the idea is to do this in people as well. You could get a shot once every quarter. It would provide protection. You wouldn't have to eat peanuts every day, which is what happens when you're desensitized. And this is now in human trials in Australia. We're going to know the answer early next year, and we're very excited to see that. So I'll stop here. Thank you for your attention. I'm happy to take any questions.
All right, well, we have time for a few questions.
Just playing devil's advocate for the exceptions to the rules that you are applying with the idea, the simple original idea that a cell is defined by the proteome, which defined by the mRNA set of, because there's people that dedicate their life to regulation at the translational level, and we also know that transcription factors can shackle between the nucleus and the cytosol and things like that. I don't know. Are those exceptions that just confirm the rule? Are those interesting things to pursue a little bit farther by some follow-up?
Yeah. No, they're very interesting questions. I mean, it all comes down to what's my view on this is it comes down to the biological question you want to ask. And if it's a question about cell type and cell state at long time scales, RNA is a great proxy for that. And I don't think you get a lot by adding protein to that. Where the proteins become absolutely crucial is as the time scales get shorter and you're interested in dynamic behavior on shorter time scales, then the RNA is definitely not sufficient and it becomes crucial to understand that. And so I think it's all a question of what time scale are you interested in. And the RNA is very well tuned to very long time scales. And those are the kinds of, as you've seen, the kinds of issues we've addressed in the talk here.
Yeah, the one further aspect, and I suppose maybe is the same answer that has to do with the time, is RNAs that are also repressed. So the same idea, right?
Yeah.
Any such thing is always answers over cell type to more daily needs for change.
Yeah. Yeah, and what else to say about it? You're okay? All right, okay, good.
And then if I can have a second one.
Yeah.
I just wonder how, the systems that you've studied are great from the point of view of your interest in human health and how to tackle it. If you were asking questions that have to do more about evolution of different cell types and things like that, will you look at simpler organisms and draw something across the tree of life that, maybe, I don't know how medically relevant it would be, but it would be.
No, 100%. And this is the motivation for why we structured the model the way we did, so you could ask those questions. And it provides a way to measure distance between cell types across evolutionary time in a way that's meaningful. Like, you know how to measure distances between genes to calculate phylogenetic relations between genes. This provides a way to do it between transcriptomes, which had been lacking. And the second paper I mentioned, the transcriptomer one, is much more concerned with these evolutionary relationships. And indeed, I think to really get the full evolutionary benefit of this approach, one needs to make cell atlases of these very early animals. I mean, and there's a ton of interesting work to do there, and that is for sure the next step in understanding the evolutionary applications and getting insight. Yeah.
Great to see evolution and expansion, the number of things we can measure through single-cell RNA sequencing. Do you think we're done measuring and sequencing cells? And now is the next before it, will spatial transcriptomics really transform our understanding of collecting more and more of these parameters, or is it time to sort of move away from that and understand what underpins these changes to other approaches where we have to perturb the system at a similar scale, which is obviously limited right now?
Yeah, great jacket, by the way. So, yeah, the question is, are we done sequencing? We have enough data. And the short answer is no. And we had built these atlases as healthy references with the notion that people would layer on top of it their measurements of different disease states. And those comparisons would be interesting and useful. So I haven't talked at all about measurement on disease tissue. I've talked a little bit about tumors, but just the very end. But that's all being done in more sophisticated ways now. And so that's continuing to fill out our understanding of the difference between health and disease. I had CZI, I kind of thought we were done. I was like, we're not going to fund this anymore. We had done the first 100 million cells. We de-risked the whole field. Other philanthropies, federal government were getting in funding. I told the program officer, congratulations, you've had an incredibly successful program. Now we're not going to do it anymore. That's your reward. And he didn't give up. And people started, in the community talking about, oh, we should do billion cell projects. A lot of it was driven by instrument companies trying to sell instruments and reagents. We had to deal with it. And, I kept saying, no, no, no. And then finally I said, okay, We can do a billion cell project, but only if it's at, what was it, 1/10 the cost of the 100 million cell. We spent $250 million on the 100 million cell project. The BioHub part was only 4 million of that, by the way. But most of it was sent out to other groups to do tissue-specific stuff. And we're not doing that again. So we do billion cells for $20 million. I'll do that. It'll be a 100-fold reduction in cost per cell. And so I threw that as a challenge, and they met the challenge. So now there's a billion cell project that sees, you can read the press release on it. By the end of next year, there should be about 10 times more cells out there in the literature. And some of it is early animal stuff to fill out the tree of life. It's kind of very interesting. He pulled together a great group of scientists to figure out how to allocate that budget of a billion cells across health, disease, other organisms, and so forth. And hopefully we're done. But the spatial stuff is also very interesting and adding a lot to it. We're working on a spatial version of Tabula Sapiens now that is proving to be very interesting, especially thinking about how the senescent cells are communicating with their neighbors, signaling, that sort of thing. Yeah?
Once we know everything, what do we do then?
We're never going to know everything. These are rough drafts, and we don't have every cell type, but they're providing insight for important biological questions, and hopefully it'll lead to new therapies and help people live longer, healthier lives. Yeah? Generative part, you mean? Yeah. Yeah. So, UCE is not generative. Transcriptomer is, not perfectly generative, but has a generative aspect. And, I think that's a super interesting aspect to it all. So if you look at the success of the protein language models, right? I mean, in some very rough sense, they explain to you the manifold of foldable proteins in sequence space. And not just the ones that exist, but the ones that could exist. And the generative part of the model is used to predict novel protein therapeutics, things like that. And, I sort of reason by analogy with the cell model in that it teaches us something about the manifold of potentially accessible cell states, either naturally occurring or those that can be engineered. And so I'm hopeful that it'll provide, a design tool for novel cellular therapies going forward. That's the hope. We'll see in the next 5 years whether that really comes to pass. That's why we set up the New York BioHub, by the way. Yeah.
You mentioned older mice. What about mice that have been, calorie restricted? Is that a way to access older mice?
Oh, yeah. So caloric restriction is a way to extend lifespan. It works very well with model organisms, not so well with primates and humans. And who would want to be that miserable anyways? You live longer, but it's a true effect, but it's complicated. So we haven't looked at it with mice. We did look at it with flies, and we never published that part of it. But, it's just, there's a lot of literature on flies, caloric restriction, their lifetime gets longer. But it's one of those devil in the detail things. Like the diet they give them for those experiments is like a very unhealthy diet. It's like eating foie gras every day. It's like the fly equivalent of foie gras. And so sure, if you eat less foie gras every day, you're probably going to live longer. And that comes to some of the challenges in interpreting the caloric restriction experiments. And so we never got to a conclusion I was comfortable. We got all the data, we looked, we saw it, and yet this issue about differences in diet became too complex an interpretation to me. Yeah. I'm sorry, could you speak up? Have we looked at very long-lived species yet? Not yet. Not yet, but it's a great one to do. There is a little work with the naked bull rat, and, that was done at the Buck Institute. There's a little bit of data out there, but we haven't done any of that ourselves. Yeah.
It's fascinating that you show data you can use the RNA-seq to deconvolute back to disease state. I wonder how sensitive it is and what can we do to improve the sensitivity, scalability, or reproducibility to detect the disease at an earlier stage.
To deconvolute the cell-free RNA mixtures?
Right.
Yeah, no, I mean, that's just super interesting sort of applied math questions there about stability of the algorithm. This is the deconvolution question. And, we kind of started out with a matrix deconvolution that was, it worked, but it was tricky. when we were done with that, there was a second paper came out from a group at Cornell who were using maximum likelihood estimates with Bayesian approaches, and that looks like very interesting as well. There are a lot of opportunities to improve that, I think. I think it's a fertile area.
Let me ask you something, and this is something I've noticed in some of the questions, and also that you mentioned, you talked about insight at some point, and so I'm curious in terms of big picture, especially this being a physics colloquium, how to think about the exercise of you throw a huge large language model to it versus how to draw insight. And I thought the example of the peanut allergy was very interesting. Mm-hmm. I mean, it led to actual molecular physical insight, whatever you want. So I'm just curious about how you see this moving forward as an exercise.
Well, both things can be true. You don't have to choose one or another. And yeah, I mean, I think the large language models are going to teach us a lot, and I'm very interested as a way of synthesizing all the data, but I think you can always go back to the raw data and ask specific questions, and nothing wrong with that. I mean, another thing that we did that I didn't talk about here was look at alternative splice variants. And, there's like a ton of putative new ones in this data because people in that field have focused on a small number of cell lines, and there's just a ton more out there that just hadn't been stumbled across yet. And there's also a lot of cell type specific usage of existing known splice. It's like a very rich thing to explore biology with by a variety of different approaches. Yeah, I mean, so it's a very deep question you're asking. So he's asking, about how are we accounting for small differences in cell state, like senescence, that are state related but not cell type identity defining? Is that sort of a reasonable summary? Yeah, and it sort of depends on the analysis we do. I mean, the changes when a cell becomes senescent are not strong enough that the identity changes, and we can see that by the differential of genes and the expert annotation and analysis. But there are subtle differences in state that you can measure through analysis like that one that emerge from the data. So there's just a lot of latent information there depending on how you approach the data, which is getting back to Hernan's thing. Well, I don't think there's just one way to look at it. We got another one. Sorry, what was the first part of the question? Okay, yeah, therapies. So, a couple things to that. I mean, coming back to the issue of senescence, there's a whole field called anti-senolytic drugs that are supposed to help you live longer by killing senescent cells. I think what we've shown here is the targets may be changing depending on which tissue the senescent cell is and what cell type it is. And so that forces you, I think, to rethink an approach to anti-senolytics. More generally, how do you make therapeutics? I mean, historically, it's been small molecules or biologics like antibodies. Cellular therapies are a third class that come on in the field of cancer for sure through CAR-T and things like that. And I think there is going to be a very powerful future of cellular therapies across many different kinds of disease going forward over the next decade. And I think we'll see the emergence of many future cellular therapies in areas that they don't use them now. Thank you.